This is Mr. Thompson, Science, Biology. Today's lecture is on the functions of DNA. DNA contains genetic instructions for the development and functions of living things. So we're looking at the development and the functions of living things. First off, in the development, we have to allow DNA to reproduce, which is meiosis, and to replicate, which is mitosis. The genetic instructions for the functions of living things are part of the central dogma of biology, which is DNA with RNA makes proteins. So DNA with RNA makes proteins. I'll have that on the screen for you in a moment here. Okay, in development, we talked briefly about this. You have meiosis and you have mitosis, and they're a little bit different. So in reproduction, which is meiosis, you're making a new organism or offspring. So whether it's a plant growing more plants, making seeds, or whether it's a single cell organism splitting and becoming more than one, or whether it's like a mammal where you have sex involved and you have an offspring. Doesn't matter, it's reproduction, and you're making new offspring. That's where meiosis takes place. Mitosis is done all the time. Replication of cells to help you grow or to replace damaged cells. So whether you're a plant or an animal, doesn't matter. If you're growing, you need to make more cells, and to make more cells, you need to have more DNA. And then if there's an injury or damage to a part of an organism, then it's going to need to also replace cells if possible. So the injury is small enough that the organism can repair itself. It's going to do that through the process of mitosis, making new cells to replace the damaged ones. Functions. The genetic instructions for the functions of living things are part of the central dogma of biology, and we talked about that being DNA, RNA, and proteins. But what does that mean? In other words, it's reading the instructions in DNA to make the various proteins that are needed to function as a living thing. There are many proteins needed to live. So whether you're a plant or an animal, or even a single cell animal, you need to make proteins for your daily existence. And this is where the central dogma of biology, DNA, RNA, proteins, comes from. So let's look at making more DNA. We can get real complicated on this. This is in both meiosis and mitosis. You need more DNA because you're making more cells. And there are many steps that you can do that are very complicated. You can look in your books, you can look on the internet to find the more complicated instructions if you want. But the main thing that is happening is the original DNA strand is unzipping, coming apart, and then more DNA is coming in, and using the complementary pairs, it is matching up and making new strands to go as the complementary side of the new DNA. So every DNA that's replicated has an old side and a new side, and we're using our complementary pairs. So adenine to thymine, there's an adenine and a thymine, and you have cytosine and guanine. So we got a couple cytosine and guanine matches up in here. So as you can see, the DNA unzips, and then each side builds a complementary strand by matching nucleotides, giving you two strands where you originally had one. So you have one original, and you get two daughter or new pieces of DNA. All right, back to our central dogma. DNA contains the instructions to make proteins. The first part is DNA to RNA, and this is called transcription. The second part is RNA to proteins, and this is called translation. So let's look at those. In transcription, what's going on is that, again, the DNA unzips, but this time, instead of matching with DNA to make a second set, it's going to match with RNA to make a complementary strand. So the RNA comes in and it matches up also G to C or guanine with cytosine. That's going to remain the same. But because RNA has uracil, the adenine is going to match with uracil, and then the thymine in the DNA will match to the adenine inside the RNA. So the messenger RNA, that's what the mRNA stands for, comes in, it goes to the unzipped portion, and it reads the different nucleotides there, makes a complementary strand using uracil instead of thymine. So when you're looking at a strand, if it has a U, then you know it is RNA because that's uracil. If it has a T, you know you're looking at DNA because that's thymine. Thymine is found only in DNA. Uracil is found only in RNA when we're talking about DNA and RNA strands. So that's transcription. Let's move on to translation. Translation takes place outside of the nucleus. So the messenger RNA leaves the nucleus. It goes to a ribosome that's inside the cytoplasm of the cell, the clear liquid substance inside the cell. At the ribosome, you have transfer RNA that comes in, and the transfer RNA is going to match up. So there we have our cytosine with guanine, guanine with cytosine, and adenine with uracil. And in that case, it's carrying an amino acid, which the three-letter abbreviation is ARG. And this is how it's going to work. So the tRNA comes in, it matches up to the messenger RNA, and that attaches the next amino acid into a growing protein. There are start and stop codons also, so that the ribosome knows when to start and when to stop an amino acid. So each one of these three-letter codes is called a codon. In your book, you'll find various codon tables that will allow you to look up what codon goes with what amino acid. And that would help you if you were trying to figure out which one's going to connect where. The next thing that happens is the transfer RNA. This part here will drop off the amino acid, and then it will go out and pick up a new amino acid, go back to another ribosome and try and match up. The messenger RNA will break apart and go back to nucleus, and then it is reused again in transcription. So messenger RNA goes back and forth between the nucleus and the ribosome, and the tRNA goes into the cytoplasm to pick up an amino acid, takes it over to the ribosome, and matches it up to the messenger RNA, drops off the amino acid into the protein, and then repeats this over and over again. So that's translation. This has been a lecture from Mr. Thompson's Science Biology, and this was on the functions of DNA.